Thank you. Manuel Corpas, Senior Lecturer at Westminster and creator of CLOBio, an open source library of agentic AI skills for bioinformatics. Today, I want to make a narrower claim than AI changes genomics. My claim is that for some genomic analysis, orchestration is becoming cheap enough that validation is becoming the binding constraint. I'm going to show you what this looks like on real data from my own family genomes and propose a falsifiable validation framework I think this department could help build. This is where this talk started. I had a folder on my external drive with 10 whole genomes. I hadn't touched it in 2 years. I knew for 4 of those samples were myself, my mom, my dad, my sister, but I didn't remember which identifier was which. The other 5 were from other pilot study participants, and I also had another one from my aunt. I asked the agent to resolve the identity within a constrained bioinformatics environment. It downloaded King, inspected the contents of each folder, it figured out the folder assembly, ran a kinship analysis, and came back with the correct family structure with confidence scores. I didn't have to worry about installation, configuration, reference genome version, or any of the usual bioinformatics overhead. That is agentic genomics. That is the shift I want to talk about today. What I mean by agentic genomics. Let me give you a precise definition. Agentic genomics is autonomous agents making runtime decisions inside domain-constrained skill libraries. The key distinction is operational. A pipeline is a fixed, direct, acyclic graph where every step is predefined. An agent has runtime decision points under constraints. Here is a concrete example. When I asked the agent to resolve sample identity, it inspected the VCF headers, found NOPLink compatible bed files, and chose King because King can work directly on VCF input. A static pipeline cannot make that decision. That is what runtime means in practice. CLOBio is the library I'll demo from. It is designed as infrastructure for science, not a product. 5 properties matter. A constrained skill library so that the agent cannot do arbitrary things. Runtime planning so that it chooses tools at execution time. Local first execution so genetic data never leaves your machine. A full provenance log so every step is auditable. And validation hooks so outputs can be checked automatically. CLOBio has currently 48 skills, 16 contributors, and is fully open source. My last count is that in the last 2 weeks, it has been cloned almost 5,000 times. And it has hundreds of forks. So it's a community that's really taking a lot of traction. The key properties for this talk are the provenance and validation hooks. Without those, none of the results I show would be auditable. Rather than listing all 48 skills, let me show you the chain this talk demonstrates. kinship check first, so verify sample identity before doing anything else. Then ancestry check, flag cross ancestry samples. Then PRS scoring against a reference distribution, which is going to be in this case the nearly 500,000 genomes project European data sets that we can freely download from the internet. Then concordance, where we compare against prior published results. That chain from raw VCFs to validated percentiles is what you are about to see. My family has been part of an open access pilot whole genome sequencing cohort for over a decade. We have jokingly called it the Corpasome because it included all of the genomes within my family, and it has been released under ethical approved conditions with a Creative Commons Zero public domain license. As part of my previous work at Cambridge Precision Medicine, I have 10 whole genomes on an external drive right now, but they are labeled with identifiers that are not very sort of identifiable in terms of who they belong to, from PT0018 to PT0010A. So there's no metadata. The point of using my family genomes is transparency, not generalizability. This is therefore not yet a general result. It is a controlled demonstration that suggests where systematic evaluation of agentic genomics should go first. The point, therefore, is not that this analysis is impossible without agents. It is that the cost of doing so correctly with full validation drops enough that we can afford to do it routinely. Before I run any polygenic scoring, I want the agent to verify the family structure. Validation first, analysis second. The agent computed kink-robust kinship coefficients for every pair of samples in my cohort on chromosome 22 alone. Chromosome 22 is adequate for kinship because kings needs only a few thousand common variants to estimate relatedness coefficients. Chromosome 22 provides roughly 10,000, well above the minimum for first degree resolution. It correctly identified my father, mother, sister, and confirmed my wife is unrelated. The parents are unrelated to each other, as expected, and it flagged one sample, PT0003A, as having consistently negative kinship with everyone, a hallmark of different ancestry, because kin returns negative values across populations boundaries. The result here is correct relational reconstruction under a logged execution trace. 5 genome-wide polygenic scores from Kira, published in 2018 in Nature Genetics, scored on my whole family whole genome sequence data, plus 498 samples from the 1000 Genomes Project European reference. I should note that the 1000 Genomes Project European subset is a convenience reference, not a definitive one. It is adequate for methodological demonstration, but the percentiles are reference relative and ancestry sensitive. A larger ancestry matched reference would tighten these estimates. Each panel is one trait. Vertical lines show family member positions. The most notable pattern is in the top left panel, which is what the next slide drills into. Here I'm showing or drilling down on coronary artery disease. Mother is at the 99th percentile, sister 97th, and I am 94th. Father at the median, 52nd percentile. Wife, unrelated and British, 13th. These family members cluster at the upper end of the reference distribution, for the same trait, all on the maternal side. This is consistent with familial aggregation under this scoring framework. But the important point here is not the trait story. It is that the agent recovered a stable, interpretable pattern that we can then try to falsify. This is a methodological demonstration, not a clinical interpretation. These percentiles are reference relative and ancestry sensitive. I should add, the agent's first attempt at scoring produced coronary artery disease percentiles that were off by roughly threefold because it treated missing from BCF positions as dosage 0 rather than homozygous reference. Prefix ran concordance with a 2022 reference paper, which was published and peer-reviewed, where I actually published these same results in a peer-reviewed journal. Benchmark was below 50%, but then when I fixed it, it's 100%, it is 100%. So 20 of 20 family score comparisons preserve rank order. That bug was caught by comparing against the published benchmark. The fix, which is reference-aware scoring via Samtug's FA indexes, is what you see now. That failure is the reason why I'm arguing for tiered validation, not despite it. Here I'm using the same family, but with 18 different coronary artery disease polygenic risk scores, all coming from the PGS catalog. And this is a reliability check, where the previous slide showed mother plus both children at the top decile of coronary artery disease using one PRS. But here, we know that there are 84 coronary artery disease polygenic risk scores in the PGS catalog as of today. I scored my whole family against 18 of them, spanning the methodological landscape. LD-PRED, LD-PRED2, PRS CSX, PRS mix, ANO-PRED, PPAS-T, GWAS only, from 64 variants to 6.6 million, and from 2018 to 2026. The headline is that mother is in the top 2% of the family across 17 out of 18 scores. The relative within family ordering is largely preserved across all 18 coronary artery disease scores. This is tier 2 cohort grade validation. The conclusion does not depend on which PRS you choose. If we compare agent versus peer-reviewed benchmark, we have the same samples, the same VCFs, same scoring weights, only missing variance handling and reference distribution differ. Despite those methodological differences, rank order is preserved across all 20 family score comparisons. And absolute differences arise from reference distributions, not from reversal of biological signal, okay? In terms of tier validation for agentic genomics, we have a tier validation framework that I like to present today. So tier 1 is when we have analytically derived ground truth, like kinship, which has a close form expectation of 0.25 for first-degree relatives. Tier 2 is when we have cohort grade, comparing against population distributions, like the 1,000 Genomes European reference. Tier 3 is the level we would need for clinical grade trust, using orthogonal standards like NIST genome in a bottle or CAP clear proficiency panels. That tier is aspirational as of today, and means that we cannot provide this in terms of clinical application. The contribution here is not the agent, it is the framework for evaluating agent generated genomic results. Agentic Genomics ships tier 1 for free and makes tier 2 routine. Tier 3 is what we should be building towards. For a growing class of routine genomic analysis, particularly those involving standardized pipelines and well-characterized data, orchestration is becoming cheaper. That makes validation, calibration, and provenance more important, and agents do not remove the need for domain expertise. They change where domain expertise is applied. The validation challenge has 4 dimensions: reproducibility, clinical safety, equity across populations, and the role of domain expertise. Each dimension needs infrastructure that does not yet exist at scale. That is increasingly where this department and the broader stats genetic community come in. This is a single control case. The open question is how broadly this holds. I should ask to be explicit about where this currently breaks. There is some ambiguous metadata, ancestry mismatch between sample and references, low quality VCS, and incompatible genome builds, which are all current failure modes. There are no edge cases. These are routine in real-world data. They require human intervention. Test it on data that you trust. Twins UK would be an obvious place to start. The cards on green show the other options if people are interested. But the core invitation is that simple. Any collaboration has to be scientifically fair and visibly reciprocal. If any method that owners want to wrap, I would welcome that. If there is an appetite, we can discuss benchmark efforts as well. So you have here the QR codes for the actual GitHub repository, for the documentation, and also here you have the QR code for my LinkedIn profile. Before I finish, I also would say that here you have the QR code for the tutorials, where you can find a way to build your first skill in 10 minutes. You'll see a lot of presentations and talks that have happened in the past couple of months, as well as a contributing guide and skill.md specification, and the materials for the hackathons that we've had. And talking about hackathons, if this is something that you are interested in, on April the 23rd, we'll be organizing at the University of Westminster a hackathon on AI agents for health, and it'll be a Claw Bio hackathon. It'll be at the University of Westminster, which is very, very central, next to the BT Tower. And we'll build new Claw Bio skills and extend existing ones. Examples will include genomics, pharmacogenomics, digital health, and beginners welcome. One of the big sort of pushes that I'm doing for Claw Bio is to actually helping people use this project as the first step towards the agentic by design scientific development. So beginners are welcome. This could be your opportunity to join the AI revolution and what I call agentic genomics. And with that, I'd like to say thank you for your attention, and I'll be happy to take any questions should you have any. Thanks very much.